blown. I am very personally excited for this next talk. I'm very honored to introduce Candace Lewis, who is going to talk us through Converging Paths to Repair, Psychedelics, and the Epigenetics of Mental Health. Please give a very warm welcome.
Hello, thank you so much for being here. Can you all hear me? Okay, wonderful. So my name is Candace Lewis. I'm an assistant professor at Arizona State University. I'm also an Alaskan. I grew up here, and I'm an alumni of UAA, so I'm very happy to be here. I'm also a lot of things. I'm a licensed therapist. I'm a mother of 3 amazingly nurturing and wild, crazy sons. And I'm a wife to another neuroscientist who also happens to be to another neuroscientist woman. So today I'm going to talk to you about results coming out of my lab about the epigenetic effects of psychedelics. And if you don't really understand what epigenetics are, don't worry, we're going to talk about that. So first we're going to go over the current state of mental health and what that is looking like in modern literature. We're going to talk about epigenetics and get a little bit more intimate about what that word means and why it's getting thrown around so much today. We're going to talk about studies coming out of my lab, the Bayer lab. We're going to talk about gene, network, and individual effects of psychedelics on DNA methylation. And we're going to go over the summary of our results and where can we go from here in the epigenetic world and in the psychedelic world. So one, in talking about mental health, this is considered one of the leading disease burdens worldwide, according to WHO. They're highly complex and multifactorial, meaning there's no single system, there's no single brain region, there's no single gene, there's no single clusters of behaviors that define mental health. We know from human neuroimaging research and animal studies that they involve altered neural structure and function. And we know from many decades of twin research and now from molecular genetic research that there is partial genetic etiology. We're going to unpack that a little bit more. However, this is the Western medical model. And this is the model in which scientists have to kind of work under for us to be able to be in the system. But everyone in this room knows that it's not that simple, and that drawing these hard lines around mental health, mental disorders doesn't work in the real world, and that's not how healing works either. So to unpack that a little bit more and just kind of looking at how we categorize mental health conditions, currently the DSM has these categories, not only these 3, but these are the 3 that I consider that my lab is most interested in. Depressive, anxiety, and substance use disorders, and each of those categories encompasses a lot of different phenotypes or behaviors. The first thing to really understand about these categories is they're highly comorbid. Comorbid means if you are diagnosed with one, you are highly likely to qualify for multiple of these diagnoses. We've known this for a long time. This is at the behavioral level. We now know from ginormous molecular genomic studies that they have shared genetic risk. After the Human Genome Project was completed, we thought, we're going to figure it all out. We're going to find the genes that make us who we are and which genes make us which disorder. And that couldn't be further from the truth. And really what we're finding is that the risk genes for any one of these disorders are also risk genes for the others because they are just, in fact, so similar. They have similar alterations across brain structure and function. Human neuroimaging studies are finding that when we compare someone with PTSD to depression, their altered brain structure and function isn't actually that different from each other. They're just different from those who do not present with mental health conditions. So we're seeing this similar neurobiology underlying all of these different classifications. They have common dysregulated physiology. Across all of these disorders, we see decreased neuroplasticity, chronic inflammation, and the neurobiology underlying a multitude of different mental processes, cognition, mood, anxiety, you name it. And what's perhaps most important, and where the field really needs to start paying more attention, and there are those of us who are trying, is that every single individual is different. This is not like diabetes or cancer, where we can really take these measurements and say, like, this is what you have, this is the underlying pathology. Any 2 people who have the same diagnoses might have completely different altered physiology. So 2 people with depression, one might have higher cortisol and the other one might have lower cortisol. And that's across all the systems. People are extraordinarily unique in the physiology underlying their behaviors. Even at the behavioral level, you cannot assume that 2 people with depression have the same behaviors. In fact, in the DSM right now, there's over 600,000 combinations on how to meet criteria for PTSD. Can you imagine trying to work in the medical model and trying to treat people because they both have PTSD, but there's just nothing similar about them? This is all pervasive. I'm not the first person to say this. I'm not the first person to know this. And yet the field is being very stubborn and moving forward and acknowledging this. What I find most important and most interesting about the similarity across these stress-related disorders is how important the aspect of experience is in predicting them. It is well known that experiences of trauma and chronic stress, including armed robbery, sexual assault, childhood trauma, and combat, these are major predictors of mental illness, and not just post-traumatic stress disorder, but the depressive anxieties and substance abuse disorders as well. But is it just trauma? And the reason I bring that up is because I get asked this all the time when I give talks, and I quite honestly find it such a boring question. When people say, but what do you define as trauma? Okay. There's so much more than the classical definitions of trauma, and right now the prevailing idea in the field is that trauma has to be a real or perceived threat of your life or someone else's. But any clinician in the room and anyone else who's taken a moment to dissect your own life experiences knows that there's so many more experiences in life that affect us deeply. They're not just a threat to our livelihood. So the way that I like to explain that is there are experiences of disconnection, and this is especially true in early development, when your brain is literally being built in response to the experiences you have. I find Maslow's hierarchy of needs is a really excellent way of describing all the ways in which we can experience disconnection. We can have disconnection from safety, and I think this is where the traditional definitions of trauma really fit in. We can also have disconnection from society, right? Things like homophobia, your sexual identity, religious trauma, microaggressions against any minoritized group. There are so many ways in which we can feel disconnection from our society that are not classical trauma. And then perhaps the one that is most difficult to explain, but probably everyone here knows what I mean, is those experiences that left us disconnected from our own self. When we grow up in a way that for whatever reason we were not allowed to fully explore and to blossom into understanding ourselves. And that's just not early life, right? That is dynamic, and that's across the lifespan. And any clinician knows that many people are coming in with these stress-related disorders, and they're not just talking about their threats to livelihood, they're talking about these experiences as well, because they deeply affect us. So that's a lot, right? So there's biology, there's experiences, and there's mental health, and it's across the whole lifespan. And how do we bring that all together? And that really was the whole, the 20th century of psychological research was that question, the nature versus nurture question. Who are we? How do we become who we are? And only recently have we been able to bridge these two large concepts together through the field of epigenetics and understanding that our experiences are altering our biology. It is not just our genetics that drive the way that your brain is built. It is our experiences that are shaping the molecular landscape of your brain. And there is now a whole large field showing that, especially in early life stress, these experiences are altering epigenetic regulation of neuroplasticity, immune function, and the neurobiology underlying mental processes. In my lab alone, we have translational models across animals and humans, identical twins, showing how early life experiences alters epigenetic regulation across these domains in stress physiology, immune function, cognition, and addiction-like behaviors. So, what do we mean by epigenetics? What are epigenetics? A short, sweet, and succinct way of explaining what epigenetics are is they are the biological regulators that determine if, when, where, and how much of a protein is made. And that epigenetic profile across all the cells of your body are unique to every single one of you. And they will continue to be dynamic across your lifespan with just age. Aging is going to change your epigenetic profile and also the experiences that you continue to accumulate. So take a moment, let's glimpse into the nucleus of the cells of your body. We all know about DNA, we know about chromosomes, and then if we unwind our chromosomes, we get this double helix. This double helix of nucleotides is what makes your genes. And this is typically what was referred to as your biology, right? Your genetic blueprint that you inherited from both of your parents that is unaltered from your experiences. However, we now know that DNA methylation, one of your most common epigenetic processes and the most studied, is dynamic. It can be altered. This DNA methylation changes depending on experiences and exposures. That DNA methylation is then altering how much of a protein is made from a gene. Those proteins are the literal building blocks of your brain structure and function. And we now know that the risk factors that have been associated with mental health for almost a century now, these risk factors are affecting DNA methylation and affecting this pathway that leads to increased behavioral symptoms. And this is fascinating. I mean, this is a huge shift in our field to be able to biologically measure and understand the mechanism by which an experience when you're 5 or 15 or 25 can have enduring, really impactful effects on your behavior 20 years later. So in my thinking, if we're starting to understand this pathway of etiology, this pathway of how experiences shape us behaviorally and physiologically, then we need to be thinking about that same pathway when we think about treatments. And that treatment could and should, and perhaps would more effectively, would be using the same pathway to lead to decreased behavioral symptoms. Which brings me to psychedelics. I don't need to review the literature for anyone in this room. We all know that anecdotally and personally and culturally, and now there's a large collection of clinical evidence, that classic psychedelics like psilocybin and the newer compounds that are now being called psychedelics like ketamine and MDMA are leading to symptom reduction. And we know that they're doing it across these categories of different mental health conditions. And what I think to me is so important about understanding this treatment modality is that it's experiential. Just like chronic stress and trauma is an experience that your body then translates into biology, so is the psychedelic experience. Treatment should be experiential, in my opinion. But because I already convinced you that we should stop thinking about disorders in categories, then another way to think about psychedelic healing instead of it's transdiagnostic is that it's trans-system. There's now still accumulating evidence that psychedelics are affecting neuroplasticity, immune function, and a plethora of neurobiology underlying mental processes. This is important. If we come to understand disorders not as distinct categories, but as like the shared biological dysregulation, then it is very promising to look at a treatment modality that is affecting these same systems across the disorders that we categorize. So then the question becomes how? How is it that trauma is affecting all of these systems? How is it that psychedelics are affecting all of these systems? And that's where epigenetics comes into play. And that is why my lab is interested in both pathways, about how our early life trauma is affecting our epigenetics and our overall well-being. And is it possible that psychedelics are affecting our molecular landscape or epigenetic profiles in the same way? And then lastly, because we are all different, because there are no two individuals with the same epigenome or the same brain or the same physiological conditions that are underlying their behaviors, it will be important to understand the psychedelic effect that is unique to each person, to understand if these treatment modalities are able to adjust one's physiology in the correct direction. If I take a psychedelic experience with high cortisol, will I come out with regulated cortisol? If I have a psychedelic experience with low cortisol, will I still come out with regulated cortisol? And this is a place that's called precision medicine. This is the future of all of medicine. It's making a lot of leeway in all the fields besides psychiatry. So the question is, do psychedelic therapies alter DNA methylation across neuroplasticity, immune, and mental processes, genes? In other words, are they directly targeting the molecular scars of early life stress or trauma? Are they altering those same pathways that lead to the symptoms in the first place? So to tell you about work coming out of my lab, I'm going to talk about 3 different studies. One, we have data from a ketamine clinical trial where the patients had either major depressive disorder or post-traumatic stress disorder. They received 6 infusions of ketamine across their treatment window, which was 2 to 3 weeks, and we measured DNA methylation in their blood. Another one was from one of MAP's, their first phase 3 MDMA trial with PTSD, which included 3 oral doses of MDMA with therapeutic assistance, and we measured DNA methylation in saliva. For these human studies, we conducted what my lab calls a brain epigenome-wide association study, or a BWAS, where instead of looking at the whole epigenome, we really focus on genes that are highly expressed in brain, since that is our organ of interest. This is a new way of analyzing epigenetic data. Typically in the field, we look at the whole epigenome. However, I feel like it's important to restrict our analyses. And then thirdly, we use an animal model of aged mice that received 2 injections of psilocybin. And for that study, we're able to look at brain tissue. We looked at bilateral hippocampus and medial prefrontal cortex and compared the epigenetic profiles across psilocybin and saline-treated mice. I won't be talking to you too much about the results from that study because there's just not enough time to go over 3 studies, but we'll briefly mention them. Okay, jumping right into the results. So what you're looking at here is called a volcano plot. Every single dot you see on there is a region of the DNA in which we are able to measure DNA methylation. Every single dot. There's, in this graph, there's about 25,000 dots. Every dot that is blue is significantly decreased methylation, and every dot that is red is significantly increased methylation. This is in the ketamine study. We found 126 genes significantly altered by ketamine. Now, that probably doesn't mean a lot to you, but this is unprecedented, especially with a small of a sample size, to find this much statistical significance in this many altered regions. It demonstrates just like the vast effects of the psychedelic treatment on the epigenome. This is the same data layout for the MDMA trial, where there we found 344 altered genes. Now, is that a difference between ketamine and MDMA, or is that a difference because in this trial they received therapeutic support? We don't know. Many more studies will have to be done to answer that question. And then in the brain tissue, which of course I'm a neuroscientist, that's exciting, we found over 3,000 genes altered in the right hippocampus of these animals. Now, what made me jump out of my seat when I saw these results compared to the 158 and 700 something in the other brain regions was there's a lot of evidence in the human neuroimaging literature, I even have a published paper on it, that shows these bilateral effects of psychedelics where we see increased function and structural changes in the right hemisphere. So when I saw this, I was like, whoa, this is crazy, I can't wait to publish this. Okay, so that's the same data from there, where we are looking at the right hippocampus and the DNA methylation change in the mice. Okay, so what does that mean, though? What does that mean? Those are just a bunch of dots on a graph that's hard to even understand what it all means. So one question we can ask with all of that data, what is the similarity? What's the convergence across ketamine and MDMA? So when we take all those altered genes, the significantly altered genes, we can chop them up into what category do they belong to? So when we look at neuroplasticity-associated genes, we see a high degree of overlap between MDMA and ketamine. Immune system genes, high degree of overlap. It's always about 50% of the ketamine total genes and of mental processes. Okay, but what does that mean? Okay, so we have a bunch of genes. Oh, you have 50 genes, you have 400 genes. What does that mean? We have really fancy network analyses we can do these days. We have really fancy computational models where you can start asking bigger questions of well, what does it mean for all of these genes to be altered? What systems are implicated in these genes? And these fancy models spit out, these are called lollipop plots. And they basically are telling you what systems are represented in the genes that were changed. I've gone ahead and these are the top 10 functional enrichments from ketamine. There was 24 significant enrichments total. And I've highlighted for you the ones that are immune related. And then I highlighted for you the ones that are mental processes related. This is really quite fascinating. It's amazing how big these effects are and how widely they spread across all of the complex systems that are already known to be implicated in mental health. Same plot for MDMA. These are your immune networks. So we had 344 significantly altered genes in MDMA, of which 46 significant functional enrichments came out. I highlighted for you the immune networks here and the mental processes here. Now, fascinating, I don't have time to go into it for this talk, but from the animal data, when we do this, everything that pops up is pure neuroplasticity. For any neuroscientists in the room, I mean, it's just beautiful. If you said, pick 10 of the top words that are related to neuroplasticity, those are your top 10 functional enrichments when we look at brain tissue. Okay, so for the functional enrichments, this is your overlap between MDMA and ketamine. What we will do as a field, what we will do as clinicians, what we will do with this information moving forward is, I don't know the answer to that yet, but I know that it's interesting and it's fun to be able to start mapping out what the molecular overlap is between these treatment modalities. Okay, now we're going to talk about individual differences. So I know this is a lot, let's just walk slowly through it. So one, just pay attention to the left real quick. This is ketamine data. Every column, do I have a pointer? Every column, you can see the columns of data, is a person. and we took all of their significantly altered genes, their significantly altered DNA methylation, and every single little row is one of those spots of DNA methylation, and this is on genes related to neuroplasticity. If it is red, they increased in methylation. If it is blue, they decreased in methylation. The stronger the color, the larger the effect, the more they changed. So you can see with your eye and moving right, the differences across people, across all these different locations of their genome. And you can see how there's group level effects, right? You can see that top chunk of blue, the bottom probably 80% is more red. We have to have that to work in modern science. We have to find a group level effect. This is how medical modern science works. But what's interesting and what's so important is to look at how the individuals do change differently. There are spots in the epigenome where many went up, but some went down, and vice versa. Some had a stronger effect there than others. To me, this is really important when we acknowledge that the physiology underlying the disorder is different across all people. We should be looking for differences in molecular and biological change as well. It's just harder to do for a lot of reasons. One, changing a whole paradigm is hard, and statistically it actually is quite difficult too. Same data on the MDMA on the right. Each column is a person. Each row is a spot on their epigenome. And you can even see even better there the differences between people and how they're changing. It's important to note, all of these people got better clinically. All of them statistically and clinically reduced symptoms in a very meaningful way. Looking at their epigenome could tell us a more and different and important story. So just quickly, we can also do this in their immune genes, where we see individual differences across ketamine and MDMA, and the DNA methylation of genes underlying mental processes. I don't know my time. So in conclusion, diverse psychedelic compounds induce convergent epigenetic effects. And importantly, these are not random epigenetic effects. They are solidly there in the regions that we know are implicated in not just trauma, but in psychiatric disorders as well. We're able to look at the unique changes per person through their psychedelic healing, and this will be very important moving forward for personalized care, safety profiles, all sorts of things. Maybe even saying, okay, let's look at what your physiology is and let's pick what psychedelic compound would be best for you. So the way that I see it is that the psychedelic science has a wonderful opportunity to think towards precision medicine approaches. This is something that the field of psychiatry as a whole was really behind on. We're still trying to catch up on it. These studies take enormous sample sizes, which is something my lab is trying to do. We've launched the Psychedelic Genome Project, in which we are trying to convince trials and clinics from around the world to have a collective protocol, collect the same type of samples, collect them at the same time points, and send them all to my lab so we can analyze them together on the same machines. And this is going to get us into precision medicine findings and clinical implications so much faster than the world of psychiatry, because they're still trying to catch up in that way. So I just wanted to briefly go back to this concept of connection in the way that I see how the typical psychedelic experiences that we associate with therapeutic response map so well onto these different levels of disconnection, in which we see, we've maybe all experienced, that the insight you have, the profound insight, can be so powerful for people who are still living in this feeling of not safety in their body, and to have that insight that like, now in this moment you are safe. From those that feel disconnected from society, the feeling of unlimited unity across time and space can be incredibly healing to finally giving us the sense of society and acceptance. And then, of course, the self, the hardest to describe, but the psychedelic experience that can lead to such feelings of bliss and contentment, and really getting to know your own self can be so healing for so many. So where to go from here? As you saw in the human trials, we measured the epigenome in peripheral samples in blood and saliva. There is still so much to understand and to unpack about what does the peripheral epigenome mean for brain. My lab is tackling that question right now in many different studies. I've already talked to you about precision medicine and how this type of data could really help us lend to navigating specific treatment strategies for specific people. But I do honor the question of who cares, right? Because if the idea is healing, and if we already know like symptoms are getting better and quality of life is getting better, then why do we need to look at molecules? Well, they're getting increasingly important for regulation, for our fight to change regulation status. Insurance companies are increasingly needing this type of data and overall improved access for people. Like, right, we live in this Western medical model world. Like, we're not going to change it. It is what it is, and people just really want to see biological data right now. So this is important for those goals. Mechanisms also inform etiology, and you have to understand etiology to understand prevention. And if you're not already thinking about it, I think if we care about treatment, we should care even more so about prevention, and we need to understand etiology to prevent, and of course treatment and safety. And then I just wanted to put a shout out to, in this field, I've been in the psychedelic world for 10 or 11 years now, and I think it's decreasing, I hope it's decreasing, but there's been this long-standing divide between people who are all about the experience, are all about the spirituality of the experience, of all of it. And then there are people who are all about the cellular biology or the molecules. And there's been this kind of a clash between them. And it saddens me, because for me there's no difference. Like, we are cellular beings. We all have a brain. And the fact that our experiences alter our spiritual and mental health at the cellular level, I don't know what's more scientific or what's more spiritual than that fact. And I really hope that that anyone, no matter which side you're coming from or lean more towards, we can really respect and understand that they feed each other, and that both the experience, the spirituality, and the molecules and the cells, it's all important. And I don't know what's more incredible than that. So thank you so much. I'd like to acknowledge everyone in my lab and all of our funding agencies and our clinical partners and my students and the humans and the mice. And then one last shout out, if you would let me. My lab is federally funded from the NIH. And if you're aware of anything going on at the federal level with slashing of grant money, I want you to maybe, I don't know, be aware, call your representative, and know that research like this is not possible without federal funding. Thank you. Sure, yeah, any questions? Yes, you two were at the same time.
I feel like a mic is always helpful.
If you were to get individualized location data from the participants in clinical trials, could you connect that to environmental exposure data and link with genetic or molecular?
Yes, I love that question. So once we get large enough sample size, absolutely that would be the moderation question, right? So depending on your epigenetic profile that you're going in with, which is, as you've learned today, totally dependent on all the experiences you've had up to that day, are the experiences you've had before your psychedelic experience affecting the way you experience it and the way your epigenome responds? I think the answer is yes, and I can't wait to have the data to run the correct models.
Thank you.
Hi. First, really great talk and amazing work. So my question is just kind of thinking about the causal relationships with the epigenetic changes. Are there examples outside of psychedelics and mental health where some data suggesting that causing a change in epigenetic expression then has an effect on mental health, so rather than just a correlational?
Yes, totally. You're absolutely right. It takes another scientist to recognize that, I didn't realize the epigenetics back to behavior, and that's a very important aspect of this. My sample sizes aren't big enough yet to run those mechanistic models, those mediation models. We have another paper published from the phase 3 PTSD MAPS clinical MDMA clinical trial, where we actually did associate the change with the behavioral outcomes. We only did it for 3 genes, candidate gene study. So we were able to show that change in DNA methylation was associated with the change in symptoms. So that's really important. It's absolutely where this needs to go. We just, we need bigger sample sizes, yeah. There was a hand here, and then I see your hand back there, too.
Thank you for that talk. That was really interesting. I'm wondering about the relationship between epigenetics and meditation, if you're aware of similar research to what you're doing with psychedelics.
Yeah, a couple different, so from Franz Waldenweiter's group in University of Zurich, they have some meditation papers out there. Well, they don't look at epigenetics, I'm sorry, but they look at meditation in the crossroads with psychedelics and the behavioral outcomes. There's some small papers out there that try to look at epigenetics. with meditation. It's hard to do, right, with the different designs of meditation and whatnot. However, I wanted to talk more about, like, I see a lot of type of stuff like this on social media, where people take the science and they extrapolate it. And I see things like, be careful your thoughts, they're going to change your DNA methylation. And who am I to say that's not happening, right? But I think it's just so important that we try to be critical thinkers and to hold space before nuance and, like, what does the data tell us? What can we conclusively say? And then also what is being drawn out further from that, like, maybe it's true, maybe it's not. Does that make sense? So, like, I would never tell someone that their thoughts don't matter and that their thoughts aren't impacting them. Like, we know that. We absolutely know that. How far your thoughts are penetrating your molecules, I can't answer that as a scientist yet. Does that help? Okay. And then the hand was way back there first? Yeah. Oh my gosh, that's like the $1 million question. That's what the field's trying to figure out. So, I mean, I can just, like, very quickly, briefly tell you what we do know and what we don't know, right? So these molecules are binding to receptors that we have really mapped out. We know what happens when we bind receptors. We know the intercellular cascades. However, psychedelics are doing things that we don't quite understand in a lot of ways. So let me explain it to you this way. Serotonin binds to the same receptor psilocybin does all the time, all day long, and I'm not tripping. Right? So different compounds can bind to the same receptor. We like to call it locks and keys, or no, like keys and locks. Yeah, whatever. They bind, and then magic happens, right? Intracellular cascades happen. One of the things that we know about a lot of intracellular cascades is that they lead to changes in gene transcription within that cell. So you just imagine, like, there's a cell, and, like, magic happens. Things are put into the nucleus, which more of this magic happens, and then new genes are expressed, new proteins are made. So if you think about a dendrite needing to grow, that's a very physical process, right? That's structure. So you need to build things. So you build more proteins, like cytoskeleton structure, like literal building blocks to make a dendrite grow. That needs new gene transcription, that needs new proteins. And neuroplasticity is so much more complex than is generally understood, I think, especially in this field. So there's just so many aspects of it. It's not just dendritic growth. There's also receptor signaling and trafficking, and there's just so much. So what is happening that leads to neuroplasticity is on so many different levels. I hope that helps.
I wonder if I can ask a question. Is that so inappropriate?
Okay.
We actually have a good amount of time, so there's actually going to be a lot of room for everyone's questions. I'm curious if there's going to be, or if you think that there's going to be a place for epigenetic research and policy making, especially once causal relationships can be made between, like, environmental racism and trauma therapy for disproportionate populations that have been marginalized and traumatized by the government for a really long time. Do you think, like, once those causal relationships are able to be made, that we're going to be able to create different kinds of reparations and policies for those populations that have had their water tainted, their lands taken, their food altered, the food deserts? Do you think that that's something that's going to be accepted into policymaking, or do you think that the science is going to be kept very separate?
That list is so long, right? And you only briefly scraped it. Like, all of the things in our world right now that are affecting people in so many deep ways and have been for so many generations. I don't think it's going to be immediate, right? I don't think the moment that we demonstrate causal relationships, there's going to be policy change. What I can say from my experience, and, like, the reason why I'm just so much into mental health and healing, but I'm so interested in the molecules because I think that we live in a world, right, for a lot of different historical reasons, we just don't believe people, right? Like, oh, you're not really, like, that didn't really affect you. Like the bootstrap culture and the get over it culture and the whatever. And for whatever reason, right now in today's day and age, people do believe biology. People believe data and molecules and cells. And so I hope that this can help shift some people's minds. If I can show them it's not just in their head, which is not a real thing, by the way. Your head is very real. Anything that's happening in there is real, I promise you. Stop letting people tell you it's not. So yeah, I hope that one day this can convince people and this can shift people towards more compassion and more understanding. I wish it didn't have to be that way, but if it helps, I hope, if it helps, then yes, then yay. But no, I don't think it's going to be immediate. I think we're far off from a societal shift and a policy shift to acknowledge these things.
Thank you for that. I share the same hope. Next question.
Just to understand your graphs better, they seem to show, some of them, an equal methylation and demethylation.
Unequal?
Equal.
Equal.
Your red and your blue.
Yeah. Within a person or across, like which way?
Well,
When
just the different substances that you put up sometimes would show equal amounts of red and blue. And I understood the
this isn't
one to be methylation and the other to be demethylation.
Yeah.
And yet symptoms were improving. So does that mean, so there's a lot of, blue is the demethylation, right? Mm-hmm. So symptoms were improving even with demethylation?
Yes. Yes, I understand your question now. Yeah, that's a question that I get a lot of times, like, okay, so wait, is it good or bad to have increased or decreased methylation? And that is such an understandable question because it's like how we're so taught to think all the time, what's up and what's down, and that's how we treat behaviors. But it is just not that simple at this level. So depending on where you are at a gene, there's many methylation marks. There's not one per gene. And an increase in methylation sometimes leads to a decrease in that protein. Increased methylation can sometimes lead to a decrease in the gene. Like the up and down can lead to up or down at both levels. And so that's not complex enough, but a gene going up or down can have an opposite effect on the same physiology. So let me try to explain that a little bit. For cortisol, cortisol is a pretty complicated pathway, right? It starts in the brain and it goes through the peripheral, and then it circulates peripherally, goes back in the brain to turn it off. There's a lot of genes in that pathway. Some of those genes, if you turned them up, is going to lead to higher cortisol levels. Some of those genes, if you turn them down, it would lead to lower cortisol levels. So the directionality in the world that I work in is never simple, and it's not, it's complicated. Try writing that in a grant. So it's really not up, down, good, bad in any level that I study, really, besides behavior. But even that probably is more nuanced. So yes, we see the complex change of directionality, and that's hard to wrap our minds around. It's not how we're taught to think. However, I think it fits quite nicely with what we really do know about mental health and are just not incorporating. Yeah. So thank you for that comment. I'll repeat it for those who didn't hear. He said, wow, like it just seemed like the momentum was going for so fast and so strong for so long with psychedelic regulation and change, and now it seems like the brakes have stopped. What happened? So I would say the scientific process and method from bench to bedside is what we call it, really from like basic science to FDA approval and to clinicians actually using a novel treatment. There's a paper that's cited all the time that it's like, what was it, like 33 years from bench to bedside. And that's the very small percentage of things that ever actually get studied that lead to clinical practice. So I think the public perception of what's been going on in the scientific realm has not ever been accurate because of how much public attention it got. So the amount of, New York Times and whatever, like there's just been so much public attention. That's not typical in the sciences. Like there's, in the science world, like we have so much drug development, and there's exciting things happening all the time, and people are doing their research, and it just goes at the same frustrating molasses speed as everything else. And that really hasn't been that different for psychedelics. Besides, we can say that, psilocybin did get breakthrough status twice for 2 different companies. That was cool. Also, I think misleading. Everyone's like, breakthrough status, it's going to happen tomorrow. It was never the case. It was never the case that it was going to happen fast. And I think there's those of us who knew it, and then there was the public. Obviously, there was the great event, right? There was the MDMA denial. There too much to unpack there. But there was clinician abuse in that trial, right? And it was a little bit swept under the rug, and I think that hurt that cause a lot. So we have a new administration with new priorities, and The data actually, so when you say it's come to a halt, it hasn't. Like Compass just released their phase 3 preliminary data. All of us in the labs are still doing our thing. Like there is still the same momentum, really. It's just not as hyped. So it's happening. It's slow. Do I think pharmaceutical companies have their hand in affecting things? Yes. Yeah. So, do I see, oh, like literal pharmaceutical people coming in and blocking science? Oh, yeah. I've always said, tell me you don't want me to study something without saying it. You don't give me money, right? If there's no funding, that is a loud, clear message that's something you're doing that doesn't want to be done. There's money now. Like it's not perfect, right? It's definitely not perfect. And I think there should be a lot more money studying psychedelics. However, so the shoulders we stand on, right? So like Franz Wildenweider from Switzerland and Johns Hopkins and New York University and UCLA, all the people that really got this going again in the 90s and 2000s, they were purely funded by private funders. There was no federal funding in those days. And then there was the hit piece on Roland about all the unethical things he did, and I think it's just such a really nuanced and complex area of science because there was no choices. Like, yeah, mistakes were made, like bad decisions were made, but when you want to study something and there's no money, there's only private money, there's going to be like the invitation for cross ethics is just it's ginormous. Now there is some money, it's coming in, so it's just all forward motion. But like there was a day where there was zero federal money in this field, and that's not that day anymore. I don't know who's next. Okay.
Hi. In terms of practical application for clinicians in the field, do you recommend any, testing DNA sites like 23andMe, or is that an underdeveloped area right now?
Totally underdeveloped right now. Yeah, we're not there yet. You can find many private companies already right now that are like, let me measure your DNA methylation and I'll tell you how healthy you are. I'm not even sure that's legit yet. But I think one day that's what I hope to help build towards, is having an understanding of one's unique biology before we make treatment decisions. But we're not there yet. I'll call you when I have a product.
We have time for about one more question.
Okay.
So it seems like we've heard a lot about the know thyself concept, where the Amanitas, we know this works, we have the data to back up this works, but you need to find yourself in that and what works for you. And now we have incredible proof, thank you so much for this information, of that many genes that are being active. Still know thyself with the medicine and how much, because we can't predict the choice factor. So when it comes to the gene expressions, are there research that has already been developed on specific markers and the application of these molecules on specific markers?
No, that has not been done. I mean, this is so you'll hear from Simon in the room today, who has some more DNA methylation data. But besides his and mine, this is the extent of DNA methylation data in humans. Yeah, see, like that is so important, and that's what I never want to get lost in my data, is that like if we blast your epigenome and we like reverse every mark of every experience you've ever had that's led to your symptoms, it is still not going to work if that person does not show up to the healing wanting it, at least curious about it, and then implement the lessons learned. It's not a panacea. It's not a silver bullet. Multiple people have said that here. This does not cast out the demons. There's the individual choice. There is the person who needs to put in the work. So that's what I always like. I don't want it to get lost in my messaging, right? It's just about this. is that you still need to consistently implement the change.
Thank you.
Thank you. I love your shawl. So fabulous.
And we love your brain.
Oh, that
can
was
we
awesome.
can we please give a huge round of
Okay.
applause for Candace?